Hi guys, this is lecture 18, Polymorphisms, and this material comes from chapter 10, page 269 to 280 in your textbook. So questions begin with, number 1, haplotypes are what? Number 2, what are microsatellites? And number 3, microsatellites are useful genetic tools in forensics because why? So far we've been talking about mutations, and in populations it may not always be obvious what the original sequence was versus the mutated sequence. So a more general way to talk about mutations is to describe them as polymorphisms. Polymorphisms in genetics are different sequence variants within a population. For example, you may have individuals that have a different base at a particular position and different alleles in different segments of the population. So these are differences that are not necessarily harmful or deleterious, just different. Some types of polymorphisms include single nucleotides, which would be equivalent to a point mutation, but we call these instead single nucleotide polymorphisms, or SNPs for short. Another example are polymorphisms which are variable in length. These are generally repeating segments of DNA, and we call these microsatellites. A third type are variations in insertions and deletions, or indels for short. Today we're going to talk about SNPs and microsatellites. I have this picture down here because there are implications for using these tools in forensics research as well as ancestry research and medical research and so on. SNPs, or single nucleotide polymorphisms, are variants within a population at a specific base. These bases could be in protein coding genes, or they could be intergenic, meaning not in a gene, and they vary between different members of a population. On the right-hand side is a diagram showing this particular position, which is an A on the top, is a G in the middle, and a T on the bottom. These three individuals would be divided into three different populations based on their SNP. This is really useful information because it can be it can essentially genetically fingerprint individuals so that they can be classified based on their genetics, not just on physical fingerprints. SNP analysis becomes really powerful when you combine it with nearby SNPs. This is called haplotype analysis. When you look at a segment of DNA, for example, this segment down here, it could be hundreds of bases long, it could be 1,000 bases long, it could be millions of bases long. But in some cases, these segments of DNA, for whatever reason, do not recombine during cell division, and haven't recombined in potentially many thousands of years or millions of years potentially. And so this region may have been inherited as a single unit from parents to offspring over many generations without being mixed up by genetic recombination. What this shows you is a segment of DNA, which could be several thousand base pairs long, but within that segment are a variety or several SNPs, which are highlighted here with circles. So there could be a G that is in this position here, a G in this position, an A, a C, and an A. These SNPs will be inherited as a single unit from parent to offspring. And so the haplotype would be, these would just be the variable bases, and so the haplotype would be GGACA. Let's look at a specific example. This type of haplotype analysis is really powerful when it's combined with some kind of phenotype. For example, eye color. Here on the left-hand side are the different eye colors, black, brown, and blue. Here in the middle are the different segments of DNA. For simplicity, we're showing a short segment of DNA, but this could be several thousand base pairs. The parts that are highlighted in different colors are the variable bases, the SNPs. Notice that in position 4 here, it's either an A or a G, depending on the person. In this position over here, it's either a G or a T. And then finally, in this position over here, it's either an A or a deletion. So a haplotype could be described as being just describing the SNPs, A, G, and a deletion for this haplotype here on top. And if you count up the people in this population, 2 out of the 6 people have that group of SNPs, or that haplotype. It's this person, number 6, and this person, number 4. And notice that person 6 and 4 both have black eyes. We say that that haplotype and those SNPs are linked to the black eye phenotype. Same deal for the next one down, individual number 5. Their haplotype G, T, A, and notice that there are 3 out of 6 people with that haplotype. Again, we say that they are linked to that haplotype is linked to the brown eye phenotype. And finally, blue, A, G, A. Only 1 out of those 6 people has that haplotype. So we can use this information to identify or predict phenotypes based on people's haplotypes. This is very powerful when combined with very large populations. Take a mixed population, shown here on the left, and sequence all of their DNA and combine it together. Then, through DNA analysis and SNP and haplotype analysis, divide them into separate groups based on their haplotypes. Often this is done in medical research to link particular groups to diseases. For example, macular degeneration was genes for macular degeneration were discovered this way, and for type 2 diabetes. So complex diseases can be understood a little bit better when we can link specific groups within a population to a haplotype and to a disease. Another type of polymorphism is a variable repeat, and this one that I'm going to talk about is called a microsatellite. Microsatellites are small segments of DNA that repeat, and the repeat unit is between 2 and 6 base pairs in length. Here is an example. The microsatellite in this example is GT. Notice that the GT sequence is repeated several times. Because this is not typically in a protein coding gene, there is typically no selection for or against this microsatellite. In that sense, microsatellites tend to fluctuate quite a bit within a population, which makes them a useful genetic tool to help sort out people within that population. Again, important for forensics, important for ancestry, important for medical science. And these microsatellites, because they are not under selection, can be longer or shorter in different individuals, with no fitness consequence to those individuals. We have 3 types of microsatellite lengths, A, B, and C, and notice that they're all the GT repeating bases. However, they are different overall lengths. You can analyze these by gel electrophoresis, where the DNA is run on a gel and analyzed by length. Notice that these underlined segments on either side would be primer binding sites. You could amplify the region that contains the microsatellite. If you were to do this for A, you would get a very heavy or long band because it's at the top of the gel. It doesn't travel very far into the gel. B is shorter microsatellite, and so it travels sort of an intermediate distance in the gel. Finally, C is the shortest. It travels the furthest in the gel. Then you can analyze individuals. Remember, everybody is diploid. We have 2 copies of every gene, and in this case, 2 copies of every microsatellite. This individual here would have 1 of the long versions and 1 of the medium versions, whereas this person in this DNA from this lane would have 1 microsatellite of the intermediate version and 1 of the short. Microsatellite analysis can be used for forensics, for medical science, for ancestry. It can also be used for paternity testing. Let's see how this works. Each one of these locus on the left-hand column is a different microsatellite which will be found somewhere in the human genome. The numbers in these rows represent the number of repeating units of that microsatellite. We don't know from this table what the actual sequence of the microsatellite is, but for this analysis, it doesn't matter. All we need to know is the number of repeating units. Everybody is diploid. You inherit one copy of your microsatellite or genes from your parent, from your mom, and one from your dad. So for each person, either John Doe, Johnny Doe, or Jane Doe, they have two versions of the microsatellite if they are heterozygous. For example, John Doe has 9 repeating units of this microsatellite CSF1PO, or 12 repeating units. In some cases, John Doe is homozygous for the microsatellite, meaning he inherited the same number of repeats from both parents. This is designated with a solid box shown here. For this microsatellite, he inherited 11 repeating units from both dad and mom. Now, how does paternity testing work? Well, you analyze the microsatellites from the dad, the alleged dad, and the microsatellites from the mom, and then from the child. Notice that the child, Johnny Doe, inherited a microsatellite that has 12 repeating units from the dad, and inherited a second copy that has 13 repeating units from the mom. That's suggestive that Johnny Doe really does belong to the alleged father, John Doe. To be sure, though, you have to analyze many microsatellites, and that's why there are many rows in this table. But you'll see, if you go through it, that in every case, you can assign microsatellites to one or the other parent. For example, in this, they're color-coded where pink is the mom and blue is the dad. All right, that is all for polymorphisms for today. I will see you in class.